come with me while I complete the Sauerkraut Fermentation Lab. Proper personal protective equipment is worn, including a lab coat, gloves, safety glasses, and secured hair for safe handling of biological samples. A clean microscope slide is labeled with the sample identification and time point. This slide represents the 0-hour sauerkraut brine sample. Here I'm showing the test tube that I collected the sample of brine in. A small drop of distilled water is placed onto the slide. This serves as a medium to help evenly distribute the bacterial sample. I used a this is what I'm doing here, sterilizing the inoculating loop. Sterilize it for 10 seconds, make sure it is bacteria free. I'm going to let the inoculating loop cool down a little bit. We are grabbing that tube of brine that was collected at the 0-hour mark. And with our sterile inoculating loop, a small amount of sauerkraut brine is added to the water drop. The sample is gently mixed within the drop and spread across the slide to evenly distribute the bacteria, creating a thin, uniform smear. This allows individual cells to be observed clearly under the microscope. The inoculating loop is then sterilized again. Here I'm showing my next slide. This was for the 12-hour mark, which I missed, so we decided to do it at the 19-hour mark when I was finally able to do so. We're just going to repeat all the same processes. Put a drop of distilled water on the slide. Sterilize the inoculating loop. Here I am grabbing my second test tube, which contains the 19-hour mark brine sample that was collected. Using the sterile inoculating loop after it's completely cooled, a small amount of sauerkraut brine is added to the water drop on the slide. The sample is gently mixed within the drop and spread across the slide to evenly distribute the bacteria. When I was finished with this, we sterilize the inoculating loop again, kill off all the bacteria. And here at the end, I am just showing where my samples are smeared.